Section on the green plants, and that section is going to cover the angiosperms. So, angiosperms are one of those groups that is absolutely massive. All right, so this is not a, and like the other groups we've seen, gymnosperms is not a taxonomic group. It's just sort of like a grouping of phyla. Angiosperms is the same way. A little bit later on, I'm going to give you the name of one of the phyla, but there are so many of them, it's really impossible to cover in a class like this. So there's like 250,000 species of flowering plants. Really, it's a massive group, right? So just like the arthropods are the biggest group of animals by far, angiosperms are the biggest group of plants by far, right? So flowering plants is what we're looking at next. And really, the key thing about the flowering plants is that the ovules, right, so the female gametophyte, is going to be completely surrounded by sporophyte tissue. In contrast to the gymnosperms, where there was no sporophyte tissue around the ovules, in this case, there actually is going to be some enclosure around the ovules, that being the ovary in this case. And because there's an ovary now, that means there has to be a flower. And if there's a flower, that means there's going to be fruits. So that's kind of the trajectory we're seeing here with the angiosperms is a progression for the enclosure of the ovule to protect it by an ovary, which then turns into a fruit. So the carpel is the part that eventually turns into the fruit, and the carpel is also a name for all the female parts of a flower. All right, so as we're going to see, the carpel is going to include the ovary, the ovules, the stigma, and the style. All right, so those last two we haven't seen before. We have talked about what an ovule is. Ovary is what surrounds the ovule. Stigma and style are the other two functional parts, female parts of the flower, that make up the carpel. So as we're going to see when it comes to flower anatomy, you could almost take an entire course in different kinds of flowers and parts of flowers and those kinds of things. But in terms of basic structures, the female parts are called the carpel, and there's also a couple of other names for it, which we'll see as we go along here. So the angiosperms are the youngest group of plants. They've only been around maybe around 200 million years, give or take. The oldest known angiosperm in the fossil records, this one is one that's extinct, is called Archaeofructus, which really just kind of means old flowering plant. Um, there's a modern day relative of Archaeofructus called Ambarella, which appears to have flowers that are very primitive, much like the ones that would have been in the fossil of Archaeofructus. It's a very primitive fossil, a living fossil kind of a plant as well. So this slide is showing us basically just the gymnosperms and angiosperms. So in the last recorded lecture, we talked about the Ginkgophyta, the Gnetophyta, the Coniferophyta, and the Psychophyta. We also have Archaeofructus, which is extinct. We have Ambarella, which is the closest living relative of Archaeofructus. And then we have a whole lot of other angiosperms, right? So a few of these are kind of like unique enough and primitive enough looking that they warrant their own groups. But then we get into dicots, we get into monocots, right? And we talked about those back in our plant form lecture. Those are, especially the monocots, the biggest groups of flowering plants. All right, so the key thing with flowers to remember is that flowers are really just a modified stem with a modified leaf. All right, so the part of the flower that attaches the flower to the stem is called the pedicel. Kind of think about the pedicel as being the flower equivalent to the petiole that we talked about with leaves back in a previous lecture. The pedicel is a little stalk that connects the flower to the stem. The pedicel connects to the flower's receptacle. So the receptacle is the point of attachment of everything else on a flower. All the parts of the flower all come together and fuse at the receptacle. And again, if you get into a plant taxonomy course where you're actually looking at identifying plants based on flower position and flower structure, you get into all kinds of detail about position of different parts relative to the receptacle and so forth. It's a really, really big topic you can get into if you really take the right course. Once you get beyond the receptacle, then there are four, or potentially four, different rings of flower parts that all attach to that receptacle that are all made up of rings, right? So whorls just means a ring of structures. So there is going to be a ring that makes up the petals, a ring that makes up sepals, a ring that makes up the male parts, and a ring that makes up the female parts. And they're all sort of arranged in that ring-like structure around the center of the flower. So in terms of the outermost part of flower parts, these are called the sepals. If you take a look at all of the sepals together, that makes up a structure called the flower's calyx. C A L Y X is calyx. So all the sepals together are the calyx, and the sepals usually are the parts of the flower that, when they're there, are going to protect the rest of the flower from damage. All right, so if you think about like a rosebud, kind of a thing, which most people are at least vaguely familiar with, when a rosebud, before the flower is opened, the bud is made up of these green leaf-like structures that enclose the rest of the flower and protect it. And those green leaf-like structures are called sepals. Right? So they are going to protect the rest of the flower when it's closed. Now, it's important to remember that not every plant produces flowers that have all four flower parts. Some do, some don't have all four flower parts. But if a plant does produce a flower that has all four types, sepals, petals, male and female, then we say those flowers are complete. Right? To have all four flower parts makes a flower complete. If a plant produces flowers that are missing one or more of the flower parts, we say the flowers are incomplete in that case. Another sort of term that you can use to describe flowers is perfect and imperfect. Perfect flowers are monoecious flowers. They're both male and female. They have both male parts and female parts. Imperfect flowers are either male or female. So in those kinds of species, they are dioecious. Okay, so the flowers that are there are either a male flower or a female flower. Anyway, back to our flower parts. Sepals are the outermost ring, usually protective. The next layer in is made up of petals. And so if you take all the petals together collectively in a flower, you have what's called the corolla, just like the car, the corolla, all the petals together. And the petals, a lot of times, are attractive. They don't necessarily have to be, but they're attractive to pollinators. So this is the part of the flower that's brightly colored. This is the part that's bright red or bright orange or bright yellow that's there to attract pollinators. Now, if a plant doesn't produce a flower with petals, or if a plant produces flowers that have really tiny, like white petals, usually that means those flowers are wind pollinated. Because the wind doesn't care what color the petals are; it's just gonna, move pollen around regardless. So it doesn't really make a whole lot of sense for a plant that's wind pollinated to make a really big, pretty, showy flower when the wind really couldn't care less about the color of the flower, right? So in those plants, the flowers are typically going to have petals that are very small, usually white, colorless kinds of color appearances. The next layer in after the petals is the androecium. Right? So the prefix andro means male. This is made up of the stamen. Right? So stamens are the male parts of a flower, and all the stamens together make up the androecium. Now the stamens are made up of two subparts. Right? So there's a filament, which is basically the stalk, and then there's the anther, which is the part at the end of the stalk that actually makes the pollen, right? Which is the male gametophyte. We'll see some pictures here in a second that will help kind of make this more, I think, relatable than just talking about the words here. The innermost layer of a plant that is complete is going to be the gynoecium. So the gynoecium and carpel, those terms are synonymous. You can use those terms interchangeably. Carpel's a bit easier to say and spell, definitely. But gynoecium is the same thing. It's all the female parts of the flower combined. All right, so the carpels then are made up of the ovary, the ovule, the stigma, and the style. And this is where the female gametophyte, aka the ovule, is going to be housed and protected and so forth. All right, so if we take a look at this flower, this is like a alstroemeria flower, something like that. And so half the flower has been removed, so you can see the other half here, the kind of the cross section of a flower here. So the gynoecium is made up of the ovary, which is the big round part here, the ovule inside of that, which is the female gametophyte, the style, which is the long stalk here, and the stigma all the way at the top, which is where pollen's going to land and eventually be sort of trapped by the female part of the flower. The androecium, or stamen, is made up of the anther at the top, which is where the pollen granules are produced, and the filament, which connects the anther down here to the receptacle at the bottom. So again, the receptacle is the place where all the parts of the flower are going to connect. So we have also our petals, right here. These light pink structures are petals. If we had all of them together, that would be the corolla. And then these green leaf-like structures are the sepals. Collectively, again, that would be the calyx. But bearing in mind, again, that all four flower parts, the carpel, the stamen, the calyx, and the corolla, those are all modified leaves. All right, so those are our basic flower parts. And again, not every flower has to have all the parts. Some flowers might not have stamens. Some flowers might not have calyx. It just depends on the species and what they produce that way. All right, so this is the toughest part of the angiosperms, is getting through their characteristic mode of reproduction, which is called double fertilization. So this is the part that takes probably the most attention on your part to understand exactly how this works, because flowering plants are a lot more complicated in how they reproduce. And in large part, this is the reason why they've been so successful, is because they have this complicated pattern of reproduction. All right, so we're going to start off with the female side of things. The male side of things actually looks a lot like the gymnosperms, but the female side of things is going to be a little bit different in one really important way. All right, so early on, we have back in the ovary a single megaspore mother cell. And that megaspore mother cell is diploid. It has 2 sets of chromosomes, but it's going to go through meiosis to produce 4 megaspores. All right, so 4 of those megaspores are produced just like any other round of meiosis. You start with 1 diploid cell, and you end up producing 4 haploid cells. The thing about this is that 3 of those 4 megaspores are going to spontaneously degenerate or disappear. They're just going to go away. They're reabsorbed by the plant, essentially. And so they go away, and then 1 megaspore is left remaining. Now, what's interesting here, and what's different from what we saw in the gymnosperms, is that 1 remaining megaspore is all of a sudden going to go through about 3 different rounds of mitosis. Right, so 1 spore makes 2, 2 makes 4, 4 makes 8. 3 rounds of mitosis means all of a sudden that 1 remaining megaspore, which is haploid, is now going to produce a total of 7 cells with 8 nuclei, which even that by itself is a little bit confusing. Well, how can you get 8 nuclei in 7 cells? Well, one of the cells has 2 nuclei in it. So what happens is that 1 remaining megaspore goes through 3 rounds of mitosis. It makes 8 cells, but 2 of the cells fuse together to form 1 big cell that has 2 nuclei in it. All right, so we have 7 cells with 8 nuclei at the end of this. And so these 2 groups of 4 cells that are produced are going to produce what's called the embryo sac. All right, and that's going to be the female gametophyte here in a second. So the other parts of this, the integument of the ovule becomes a seed coat. A seed coat also contains the micropyle, which is going to be the position at which the pollen tube eventually is going to enter into this and deposit sperm cells to fertilize and then fuse with 2 of the structures inside of the ovule. All right, so we said on the last slide, the embryo sac is the female gametophyte. It's made up of 8 nuclei that are going to comprise 7 cells. So remember, one of the cells has 2 nuclei because originally it was 2 cells. We have 8 cells here. Now, this is where you really got to kind of pay attention for a second. So in this 7-celled, 8-nucleus embryo sac, which is the female gametophyte. All right, so there are going to be, I'm trying to see if I can find it. Doesn't really show too well on this picture. Okay, so essentially inside of this embryo sac, there is an egg. All right, so that's cell number 1. Actually, you know what? I'm going to do something a bit different here because I don't like how that picture is showing that. So what I'm going to do is I'm just going to open up a Word document real quick here, and we're going to type this out real quick. All right, this is a little bit unusual from what I normally do, but I think this will actually work better. All right, so the embryo sac is made up of 7 cells and 8 nuclei. All right, so what we have here is the egg. We have the central cell with the 2 polar nuclei. So this is that one that's a little bit strange. The central cell is actually 2 cells that fuse together, which is the reason why then it has the 2 nuclei inside of it. And this central cell and the egg cell are usually more or less right together in the center of the embryo sac. There are going to be 2 synergids, and the 2 synergids are essentially going to be around the micropyle. And then we're going to have 3 polar nuclei at the opposite end. Now these polar nuclei, I'm sorry, polar nuclei. These are interpital cells up here. I was looking at the top while I was writing here. Can't do more than one thing at a time. Okay, so 3 interpital cells are the end opposite the micropyle. So we have the egg, that's 1 cell. Central cell is 2. The 2 synergids is 4, and the 3 interpital cells is the 7 cells we talked about. The egg has 1 nucleus. There's 2, 3, 4, 5, 6, 7, and 8 nuclei there. So this entire thing is the embryo sac. The egg is what's fertilized. The central cell is going to become the triploid endosperm. All right, so triploid is just like haploid and diploid. It just means it has three sets of chromosomes, and it really kind of makes sense because we're going to have a sperm cell, and with one set of one set of chromosomes, and then the polar nuclei that have two sets of chromosomes. So 2 + 1 is three sets of chromosomes total. The synergids, one of these cells will be broken as sperm cells are deposited by the pollen tube. Can't type today. And then the other one will essentially degenerate. And the antipodal cells, these are essentially vestigial, so you don't even really need them. They're just sort of the remnants of the other cells that are not the rest of the embryo sac. And actually experiments have been done where they've gone in and removed the antipodal cells from an embryo sac, and everything develops perfectly fine even without the antipodal cells. So they're really just kind of extraneous extra cells that aren't really needed, and they don't really do anything when it comes to fertilization. They're just sort of there, and then they degenerate eventually after that. So that's the embryo sac. Hopefully this helps a little bit. I don't know, maybe more confusing, but the embryo sac is made up of an egg, the central cell, which is right near the egg, and the central cell has the two polar nuclei because that was one cell that fused into two. We have the two synergids, which sort of surround the micropyle, sort of like a block the opening to the outside world that the pollen tube is going to grow through and drop off the sperm cells. And three antipodal cells, which are the opposite end of the embryo sac, which are completely not used for anything as far as we know. All right, so that's kind of the quick rundown of the embryo sac, right? So we've got everything on this slide that we talked about, the polar nuclei, the central cell, the egg, the two synergids, and the three antipodal cells that don't have a function. Now, looking at the life cycle here, and we're going to track through this and look at how this sort of works and relate it to other kinds of plants we've talked about as well. I think what I want to do here is start down here with the zygote. No, I don't. I want to start up here with the flower because that has something that we haven't talked about yet. We're going to start with the flower up here. So the flower has the ovary, the ovule, the stigma, the style, the anther, the filament, the sepals, the petals, everything is there. But we want to focus on initially what's going on with the anther. So the anthers contain what are called microsporangia, and the microsporangia are what produce microspore mother cells. Microspore mother cells are diploid. They are going to go through meiosis, and they are going to give rise to microspores. Microspores are going to be haploid. They are going to differentiate into pollen. Right? So they're now haploid because they went through meiosis. On the other hand, up here in the ovary, we have the megaspore mother cell that goes through meiosis to make 4 megaspores. 3 of those break down. 1 is left behind. The 1 remaining megaspore goes through mitosis 3 times to make an embryo sac that has 3 synergids, the central cell with the polar nuclei, the egg, and the 2 synergids down here. Antipodals, central cell with polar nuclei, egg, and the 2 synergids down here. Now, what eventually happens is these pollen granules are going to contain 2 cells. They're going to contain a tube cell, and they're going to contain a generative cell. The tube cell is what makes a pollen tube. The generative cell is what goes through mitosis to make 2 sperm cells. All right, so eventually what happens is when the pollen lands on the stigma, the pollen tube grows down from the stigma through the style, all the way around the ovary, and up through the micropyle here where one of those 2 synergid cells is broken, and these 2 sperm cells are dropped off inside the embryo sac. So one of those 2 sperm cells then fuses with the egg to form a zygote. The other sperm cell fuses with the central cell to form that triploid structure called the endosperm. So again, 3 sets of chromosomes, 1 from the sperm, 2 from the central cell, 3 sets of chromosomes total. That will then become what's called the endosperm. So after fertilization, what we call double fertilization, we have the endosperm, which is essentially all of the energy this plant's ever going to have before it germinates or grows. This is energy. Endosperm is just stored up energy. And the zygote, which becomes the embryo. So eventually, and this is something we're going to see coming up here in the next chapter, is the development of this process where this zygote will then start to go through mitosis. It's going to make an embryo. As it makes the embryo, it uses up the energy in the endosperm. Eventually, if the seed is lucky, it can germinate. It can start to grow into a new sporophyte. And that new sporophyte eventually will make flowers and then restart the cycle again from there. All right, so the key part of this that's a little bit unusual and unique to the angiosperms is this double fertilization thing. So I'm going to go ahead and go back through this one more time. All right, that last, basically everything over here on the gametophyte side of the coin. So microspore mother cells are in the anthers. They go through meiosis to make microspores. Microspores make pollen. Pollen is then made up of that tube cell and generative cell. The tube cell makes a pollen tube. Generative cell makes the sperm cells. All right. Now, over on the female side of things, we have the ovule inside the ovary. It goes through meiosis to make, well, the megaspore mother cell goes through meiosis to make 4 megaspores. 3 of those break down, 1 is left behind. That 1 megaspore that's left behind goes through mitosis 3 times to make the embryo sac with the 3 antipodal cells, the 1 central cell with the 2 polar nuclei, the egg and 2 synergids. That's the embryo sac. So when pollination occurs, when pollen lands on the stigma, the pollen tube goes down the style, around the ovary, and up through the micropyle where one of the 2 synergid cells is broken, and these 2 sperm cells are dropped off, where one of them fuses with the central cell to make the endosperm, the other one fuses with the egg to make the zygote. And that's going to restart our sporophyte generation over here. Okay, so that's one of those things where you got to spend a little bit of time with it, maybe draw it out. Definitely look at some pictures, maybe a few other animations or lectures on the topic, because this is a really important thing for angiosperms. Probably the reason that's one of the reasons it's given them the most success on land since their appearance about 200 million years ago. So pollen production actually is very similar to what we saw in gymnosperms. It's not really anything too different. The big difference is what happens with the female gametophyte and then embryo sac thing. This is all stuff that we said on that last slide. Pollination is what occurs when a pollen granule lands on a stigma. Pollination and fertilization are not the same thing. Pollination is just mechanically pollen landing on a stigma, whereas fertilization actually involves that fusion of sperm cell nucleus with egg cell nucleus to make a zygote. So just because pollination occurs doesn't necessarily mean that fertilization will occur. It might, but not necessarily. Excuse me. We talked about the pollen tube growing down the style. We talked about the 2 different cells there, the generative cell that makes the sperm cells, the tube cell that makes the pollen tube. These sperm cells, again, are not flagellated, so they don't need water. Again, these are like gymnosperms where water is not required for reproduction because the sperm cells don't have to swim. They are dropped off by the pollen tube directly sort of on the doorstep of the egg cell so that fertilization can occur very easily at that point. And again, the idea of double fertilization is where we have one sperm cell fusing with the egg to make the zygote. The other sperm cell fuses with the 2 polar nuclei to form the endosperm. The endosperm is then the nutrients not only for the plant, but for a lot of humans as well. The endosperm is actually what we consume with any sort of grain crop that we eat. We are consuming endosperm. So corn, the starchy part of the inside of a corn kernel, the white fluffy part of popcorn, right? That's endosperm. The inside of wheat is endosperm. The inside, the rice, white fluffy part of rice, is essentially endosperm. So this is a really important thing for the plants because it is energy for them to give their offspring a shot at survival before they can start to grow on their own. But humans have also learned to take advantage of the endosperm because it is so nutritious to meet our own caloric demands as well. So it's really important for supporting humans as well as kind of a grain crops, basically. All right, so that's the end of the green plants. We're going to wrap this section up here, and we'll start up with our next chapter here in just a second.